Positive selection acts on a repertoire of receptors with inherent specificity for MHC molecules. Positive selection acts on a repertoire of receptors whose specificity is determined by a combination of germline gene segments and junctional regions whose diversity is randomly created as the genes rearrange. The unselected receptor repertoire must be capable of recognizing all of the hundreds of different allelic variants of MHC molecules present in the population, as the genes for the T cell receptor, alpha and beta chains, segregate in the population independently from those of the MHC. If the binding specificity of the unselected repertoire were completely random, only a very small proportion of thymocytes would be expected to recognize an MHC molecule. However, it seems that the variable CDR1 and CDR2 loops of both chains of the T cell receptor, which are encoded within the germline V gene segments, give the T cell receptor an intrinsic specificity for MHC molecules. This is evident from the way these two regions contact MHC molecules in crystal structures. An inherent specificity for MHC molecules has also been shown by examining mature T cells that represent an unselected repertoire of receptors. Such T cells can be generated in fetal thymic organ cultures using thymuses that do not express either MHC class I or MHC class II molecules by substituting binding of anti-beta chain antibodies and anti-CD4 antibodies for the receptor engagement responsible for normal positive selection. When the reactivity of these antibody-selected CD4 T cells is tested, roughly 5% are able to respond to any one MHC class II genotype, and because they developed without selection by MHC molecules, this must reflect a specificity inherent in the germline V gene segments. This germline encoded specificity for MHC should significantly increase the proportion of receptors that can be positively selected in any one individual. And that's positive selection acts on a repertoire of receptors with inherent specificity for MHC molecules. Have a great day.